Okay.
Oh.
Can I go like this? Okay, then I think Ben will start. We'll do this together. A bit on general good scientific practice and then a bit more documentation in between. So as I said, if you have any questions, let us know.
Also, good morning from me. So the title is clear, I guess. It's actually an kind of a workshop that we are giving for the last 2-3 years to a lot of different, let's say, scientists from different levels. I'll come to that in a minute. So to start off, what is actually good scientific practice? It's actually the principles and values and also the standards of behavior and practice in science. That's of course a very broad definition of it. In the late 90s, the DFG has made this first paper on recommendations for safeguarding good scientific practice and has made some newer formats in 2013 and also lately. Also, since a couple of years, there's a full English version available. It's a very let's say, elaborated work and an elaborated booklet now these days. So me, and I think also Martina, have not really read it, so you can go through it, and if you have specific questions, you can find a lot of things, but it's super complicated. And that's actually the reason why the universities, and that is of course given by the DFG, have also made for themselves a kind of guideline for safeguarding good scientific practice. As you can see here on the left-hand side from the TU Dresden, but every university actually. And when we give these workshops for other SFBs or SPPs, I try to find also the good scientific practice pamphlets for these different universities, and you will find them all there. This is more, it's a shorter version, much clearer than the ones from the DFG, with also practical guidelines. They are freely available, of course, and some of them we will discuss today in this workshop. So beyond the level of Germany, of course, was discussed or is discussed many times on different occasions, different places. There are different statements or different guidelines discussed in Singapore, Montreal, and they also are called then regarding the place where they have been discussed. So there are Singapore statements, Montreal statements, and based on these statements, also the DFG has developed their recommendations. And then, of course, also the universities. Also from the NIH, there are recommendations there. These are the ones that Martina, for instance, already has seen and read. I haven't been into that, but they are actually rather straightforward and quite clear. But I think also the ones from the TU Dresden are a very good guide actually for that, what we are doing or what we should know as good scientific practice. So there are a few clear lines that I actually took out of the guidelines from the TU Dresden. So they might be a bit abstract, but we'll try to explain them further on. So one of the things that is very important is that in good scientific practice, of course, is related directly to the rule or to the discipline that you are doing. So the science that we are doing. It could be like, depending on medical science or chemistry or anything else, they have every, let's say, science or discipline has its own type of rules. And that is, of course, important. Not every specific rule is directly related to a specific discipline. So documenting of data is very important in good scientific practice, and we will come to that later. They have to be comprehensible, verifiable, and of course complete. One of the, let's say, critical things that we as scientists, of course, always need to do, and that is one of the most fundamental principles in good scientific practice, is questioning or critically and consistently questioning the data, our data, other ones' data, but of course also definitely our data. Maintaining a strict integrity is very important with regard to co-workers, your collaborators, but also competitors and predecessors. So it's not only this integrity is of course necessary at any different level. Maintaining ethical standards is of course also very important when you carry out studies, but also reviewing other people's data or even in examinations. And that is actually the reason why when we were developing workshops for good scientific practice, we made actually 3 different workshops specifically for a group of scientists that need specific, let's say, recommendations to that, especially the ones for PhD students and also technicians. They are more concentrated on establishing correct information, how to set experiments, how to use specific tools or specific, let's see, materials. Is there any question? I thought there was something. So, of course, evaluation of your own experiments. What are correct controls, positive, negative controls? How do I evaluate my results, statistics? Do that. What kind of statistic test do I use for specific experiments and results? And of course, also safeguarding of your data and storing primary data. So on lab books, on servers. We will come to that in a minute. So that was in the different workshops that we gave the last couple of years, our main aim in that sense. For postdocs, of course, partially still evaluating findings in experiments, their own experiments, their own results, but also the ones from their juniors or the ones from the people that they are guiding, also rules and, let's say, recommendations how to supervise younger people. And then, of course, also safeguarding and storing primary data as for PhD students. And then we had also a workshop specifically for, let's say, senior postdocs and group leaders and professors, where, of course, evaluation of data of co-workers, of your PhD students or anything like that, but also of exams or of grants, or you as an evaluator of grants of others, how to keep their good scientific practice, also the integrity there. I mean, we have enough examples, and probably also you, on wrong good scientific practice or bad scientific practice related to reviewing. And this is, of course, part of this integrity that really is a very important pillar of these workshops. Of course, also supervision, how to supervise junior researchers is a very important issue there. And cooperations, how reliable, or what is your reliability, quality assurance. So if you are doing specific experiments for other people and you guarantee that, or you tell your, let's say, your collaborators, this is really experiments and these are results that are done according to guidelines, that these people can also be convinced and that they can be safe in publishing these data together with the data that they have. Right, so this is, of course, crucially important. So these 3 different good scientific practice workshops, something that is something that we have been doing. The one from today is more related to PhD students and senior PhD students, and also, of course, technicians in this regard. So you have seen in bold, probably in the text before, the word data. So first of all, and maybe a kind of a trivial thing maybe for you, is what is actually data? Although it's actually not that trivial. And at the time where we were doing these workshops and actually learning what good scientific practice was and how we could also bring this message to you guys, we have made actually a board, or like kind of a pamphlet here, where everyone could actually write down what they thought the data could be. And as you can see from this, data is not only a result of a certain experiment like a PCR or the analysis of a measurement, but data is as actually everything that we produce and that we make or that is made by others in our, let's say, scientific environment. This could be mice, so a knockout mouse is data on its own, You also have databases that are that you use from somewhere. This is also, of course, obviously data, but also an email, for instance, is data. You send or you receive an email with a certain result or an ID from someone else, or from you send yourself an email with an ID or a kind of a hypothesis on one or other thing. This is also data. And this is important because we're going to describe, or we're going to discuss how you have to, or how you can treat your data to preserve that, what we have to do with these data to make sure that on a later time point, when you publish data, or when there is a kind of a discussion on how to use data, or when people leave the lab, whether they can use this data for another purpose, or for another research, let's say, postdoc in another lab, how we can take this data with us, or how we can actually treat these data in the future. This is now the first step to what Martina will discuss here, how to handle this type of data.
Okay, and then we will come to the documentation part a little bit more. So yeah, Ben already alluded to what kind of data we can possess. How to handle them. There are these FAIR principles that mean that data should be findable, accessible, interoperable, and reusable. And what does this mean more concretely? So practically findable, it has to be, let's say, described in such a way, the file, that you would find the data again. So if you, for example, write PCR1, PCR2 as file names, you won't know later on what it is, and it will be very hard to find like a specific data set of PCR data that you're looking for if you were just to like have such a general term. So metadata is very important, and we will also go into this a little bit into more detail later. It has to be accessible. That means also that humans, but also especially machines, need to be able to read it. So you have to think about the data formats that you use, and also, let's say, think about long-term storage options. And also the repository that you deposit your data is very crucial. So it could be a local one, it could be a cloud. So these are also things you have to think about, where to deposit your data, especially for the long run. Interoperable is also very important if you think about yeah, using languages. If you think you work together in an EU project, everyone has their own language. It should be a language that everyone understands. And I mean, it's one example. When we started out in our lab, we were mostly German-speaking people. So, of course, you know, you're lazy, you use your own language, you write stuff in German. But then, you know, as more international people come along, the problem occurs that people don't know what it means. So, yeah, if possible, I think it would be useful to start writing everything in English. And yeah, making that effort, especially for later on, again, for everyone to understand it. But that doesn't only apply to languages, but also, of course, to formats for computers. So to make sure that you have some kind of files that every computer can read, no matter if it's a Mac or a PC or some maybe in the future computer system that we don't even know of yet. And then the data also have to be, or should be, reusable. And that also means looking into copyright licenses, knowing where they come from, because that also can be important, especially when you think of like patents or something. You have to be able to know who was the first, where did the data come from, where was it documented the first time. And especially also the licenses. Once, for example, you publish your data in a journal, then often you have to give them, you know, the rights to your data. So these are things you also have to think about when storing or handing over your rights of your data to a different journal, for example. So I think data management is easily said, but not very easy to do. There are 7 major challenges that appear to be true, I think, for a lot of labs, and we will go through them just in terms of best practices. So we don't have enough time to really go into detail of each one of them, but we will discuss at least some best practices, how it could be done to make it easier to find data again. And those 7 challenges are responsibility, data management plan, file management and naming, metadata, backup and security, ownership and retention, and then long-term planning. So coming to responsibility, it is useful to really define roles and responsibilities for people who take care of data management. So for example, in our lab, we were just in the process recently of making a data management plan because that is required from, for example, the European Union. I would imagine that maybe in the future also the countries will make it obligatory, and if you look into the new guidelines of how to write a project grant for the DFG, they also have a lot more expanded on data management than it was like 2 years ago. So this is definitely a topic that is still upcoming. And I don't want to say it's a full-time job. It could be a full-time job, I think, but it's definitely good to assign some responsibilities for people to take care of it and to at least do some initial thinking and structuring of data management plans in general. Then it can be useful to develop training plans for continuity, and that doesn't only mean really giving people who come into the lab newly a real plan of how data are handled in the lab, but as Ben already said, also for when people leave the lab, like how are they supposed to leave the data, what data can they take them with them to the new institution, etc. So this is really from entering until leaving again, very important. And then, of course, someone also should monitor the results, because the whole documentation only makes sense if people really feed into the documentation plans and really write down what they've done. So also this would require some monitoring. So coming to this data management plan, this is actually a very comprehensive file in the end of stuff that you should reconsider in how to acquire, handle, store data, and also about ownership. So best practices, what should be included in there. For example, what types of data will be created. So that could be, for example, PCR files. It could be microsite images or something where you have, again, very specific data files. So you can mention, for example, all different kind of file formats that you will collect and try to make sure that everyone will have access to them. So, if your collaborator in Denmark wants to read your microCT data or something, then they also have to find a way that they can also access it. Then also ownership is an important topic. Who will own the data, have access to the data, especially important if you use human samples that have to be encrypted, and maybe only a certain number of people should be allowed to see those data at all. So that has to be defined. And then also who's responsible for managing the data. Then another point is what equipment and methods will be used to capture and process the data, and that again a little bit, I think, goes in hand with what types of data files you will generate. And then also a big important topic, where should data be stored? And again, maybe also coming to the, let's say, readability of, either having a language again that is consistent, and that of course goes also in hand with, if you think about US English and German language, where you use the commas and the dots and numbers for different meanings. So those are very small aspects that make a huge difference. So all these things should be defined beforehand and should be agreed upon in which way you display the data. Okay, file management and naming. So I think this is maybe one of the most critical aspects of really long-term storage of data. And I don't know if you all have your own folder structure somewhere and how you structure your files, but maybe this sounds familiar to you, maybe it doesn't, but for our lab, for surely it does, that everyone has their own structure, and that in the end leads to that you have inconsistently labeled files. For example, you have multiple versions. Let's say you write a paper and then there's version 1, 2, 3, 4, 5, 6. But maybe it's not even written like that, but in a different way, and then it's unclear which one is the latest version. You could have inside poorly structured folders. It could be stored in multiple media in several locations in various formats. So I think there's no end to these examples of how different folder structures can be. And I think one of the main aims is really to organize this in a way that some outstanding person who did not generate those data can actually find the data. So best practices for file naming is to avoid special characters, because again, in German we have these Ö and E, like with these double points up there that maybe English, English language doesn't have. So I think every language has their own special characters, so don't use them if possible. Use capital letters and underscores because it makes it easier to in search platforms. Of course, the name should be kind of limited and not be too long. You should use documented and standardized descriptive information about the project. If possible, for example, the date is another huge thing that everyone writes differently in different countries. So also here it would be useful to agree on one format that you use consistently. And in general, it's good to include version numbers if you, again, have a standard way of defining what the latest version is. Okay, and in line, I think, with file naming is then the metadata, because that is structured information that describes, locates, or makes it easier to retrieve and use an information resource. So it sounds a bit complicated, but practically that means how will someone else be able to make sense of your data, and especially if you think of an Excel sheet of the cells and values that are in your spreadsheet. So for you, for example, it might be intuitive if you have let's say everyone works maybe with bone here and you have your most common micro-CT parameters and you just write BVTV, but someone who works in cardiology and maybe enters the lab will not know what that means. So it's important that at some point you say actually what that is, and also maybe define the units so people later on who are maybe not experts in that know what you're talking about. Exactly, then universal standards, again, to label data and how to describe a data set to make it discoverable. And best practices here are, for example, describe the contents of the data file, define parameters and units, explain formats for dates, time, geographic coordinates, so this we already had too. Define any coded values, define also quality flags or qualifying values. So I think actually as one good example, whoever works with ABI real-time PCR machines. So there, for example, they always have these flags. If one PCR value looks weird, then they make this triangle with a 1 or a exclamation mark, and then later they say what the problem was, or like was it below the threshold or something. So I think that's a nice example of how they make flags and then also explain what it means. And then also very important, define missing values, because sometimes you get a data table, there's no value for one mouse, let's say, and you were wondering, was this mouse not included? Did it die beforehand? Was that just not measured? So it's hard to know afterwards if it's just empty, what that actually means. So here's one example of data that you could collect and the metadata file. Maybe this makes it easier to understand. I think it sounds quite complicated in theory, but it's actually not that complicated. So here, for example, you could have an employee ID, the first name, the last name, and then here it already starts NIN, whatever that means, and then department ID. And then if you would go to the metadata file, you could say, okay, employment ID, first name, last name, and then NIN, here it's described, it's the national identification number. Let's say for gender, you could then define, okay, there will be male, female, and null. So this is a very nice example of a metadata file that explains everything that you've used in your data table. Okay, coming to backup and security, very important to make copies of your work. So if your computer crashes, you still have your data available. And the best practice is actually to make 3 copies. So to have the originals, to have an external local copy, and also an external remote copy. So worst case scenarios, the building, I don't know, burns down or something, then if you would have a remote external copy, you would still have your data. Whereas, if the building burns down, then even your, local external copy may be destroyed. Where to save it, either on hard drives, on tapes, on cloud storage. I think it's still important also to keep hard copies. So yeah, there are lots of different things, and I think depending on the institute that you work in, there may be even systems available, and again, regulations where you can save it and where you cannot save it, especially when it comes again to human data. There have to be maybe more secure deposits than like if it's for your own personal data, you may not care so much about it if someone in the US could potentially read it. But I think especially at the medical faculty here, we have quite strict rules about this. It is also best to back up your data unencrypted. But again, in the case of human subjects, that's not always possible. And in that case, you should keep the passwords and keys on paper in 2 copies and put it in a PGP, which means a pretty good privacy encrypted digital file. So coming to ownership and retention, the question is, how long do I have to keep the data? How long is long enough? And for that, we just want to show this one example of Retraction Watch, where a paper was published some years ago, and they had to retract it in the end because the authors couldn't find the original data anymore. And I mean, you know, these are things that can happen even if the paper was published like 13 years ago. It went through peer review. Several people read it in the meantime, and it was cited heavily, actually, this paper. But still, at some point, someone finds, I think in this case, it was about also blots, or no, it was about the duplication of a table or something, and they couldn't, you know, if you then can't provide the original data, then the journal was forced like to retract the paper. So yeah, depending, you know, on what you're thinking about here, you have to try to be able to retrieve the original data for as long as possible. However, there are policies, of course, that you can or you have to actually apply to. So for example, in Germany, the DFG, if you have a DFG grant, for example, they require you to keep the data for 10 years. NIH, for example, for 7 years. If you work with S2 organism, you have to keep your documents for 30 years. But it may also depend on different policies, for example, intellectual property. So if you want to, you know, file something for a patent, you might have to keep it even longer. There are IRB data retention policies. Then from, as I said, the funders, like the DFG has their own rules. Publishers have their own rules, and also federal and state laws have their own rules. So you really should know what rules you have to abide to and stick to them. Then coming to the ownership question, and as for everything, I think in good scientific practice, there's never a straight answer. And also in this case, it's not so straightforward. Again, in Germany, at universities, we have the so-called freedom of science, which per se means that the owner of the data is the person who created them. At a university, there's only one exception if you make an invention, because that always automatically belongs mostly to the university. I think 70%, right? And then only 30% belong to the real inventors who had the idea. But I mean, okay, I think inventions is, let's say, a more or less common thing in our field. So we want to publish our data usually. And in that case, there is this exception that may be defined in your contract or not. I mean, this is also something maybe specific that you have to inquire for yourself if this applies to you. But there is this exception that if you perform research within a duty, for example, you come in as a PhD student, you are assigned a DFG project that you have to work on, then the data actually belong to the institution, not even to the PI, because usually the DFG, for example, gives the money for the research to the institution. And even though the PI at the moment he's there can, let's say, has freedom of conducting the project as he sees fit and using the materials as he sees fit, or she, let's say if the PI was supposed to go to a different university when the project is still running, the PI also would have to negotiate with the institution if he or she can take the data with him. So it's not that straightforward, and it always has to be discussed. So I think this is really the main, let's say, take-home message from this slide, is that if possible, you should always discuss the ownership before the project starts, because usually also there's not just one person collecting data, but several. So this is also crucial then if you think later about publishing a paper, who gets to be in what position. So I think if possible, discuss this beforehand and define who can take what data with him if they decide to leave, for example, in the worst, not in the worst case, but in a final case. And because publishing and authorship is a really big and important topic for everyone, this is also one of the aspects where it comes to a lot of disagreement. And if there are some disagreements or conflicts you can solve on by yourself, there's this person of trust, the ombudsman, that is also at university level. Again, I think there's like the own society for good scientific practice, and they also have like a national ombudsman or something like that, right? Yeah. So there are different levels if you wouldn't even feel comfortable to discuss that at your own university. But I think usually they provide, so I think there's still the slide here from Ben, where you see the ombudsperson of, let's say in our case, the School of Medicine. Again, depending on where you work, the TU Dresden also has their own ombudsmänner. So maybe there's someone who you know could be impartial, and then you can discuss with them. I think, so yeah, okay, let's point long-term planning. Again, this is what we already briefly discussed. When choosing file formats, be sure that it is a consistent format then can be read well into the future. So what does that mean? I don't know how old you people are on average, but if you remember maybe in the, I guess, 80s, early 90s, there were the floppy disks. So that, of course, at some point became replaced by CD-ROMs and then later DVDs, and now everyone has USB sticks. And no computer these days anymore could read a floppy disk. So these are things, yeah, you can't predict, of course, what's going to happen in the future, but I think if you already notice over time that some medium might become obsolete in the future, I think it maybe pays off to transfer these data when you still can onto a different medium before it gets obsolete and no one can read it anymore. And then again, for our non-proprietary data, it should be open accessible, documented standard, unencrypted again, as we said, if possible, uncompressed if possible. As you know, we also collect lots of data these days with these sequencing analysis. So yeah, of course, sometimes compression is more useful, but best practice would be to do it uncompressed. Then there's this American Standard Code for information that is really readable by all computers. So that is, for example, a format that is readable also well into the future. Okay, yeah, that was it from my side. I think then Ben will take over with lab book.
Yeah, so a little bit more concrete and to one of the things that, of course, is for all of us important, at least at one of the stages, especially when you are a starting PhD student or technician, is how to actually collect or how to put your data into kind of a format that it is, of course, later by others and also by yourself readable and verifiable. So the purpose and the disposition of lab notebooks, to start with that. And the reason why we actually added that to this workshop is because we encountered that a lot of people do not really know how to handle and how to fill in lab books, notebooks. So, and what are really the needs and the rules that belong to notebooks. So, your notebook is an individual lab notebook that belongs to you, right? And it provides a complete and a permanent record, and that is very important, of all the aspects of your experiments, but also of thoughts, ideas, hypotheses that you have or that you have discussed with others, with your group leader, or anything that you think is of the importance to keep, right? So your lab notebooks belongs to the institute. This means that your lab notebook will also never leave the lab at any time, but you are allowed and you're actually encouraged to copy pieces or parts of your lab book so that you can take them with you when you go, for instance, write a paper or anything at home or anywhere else, to be able to use this information on a later time point. What is now the lab book itself? It might be sounding, or it might be trivial for some of you, but actually a lab book at first, and later Martina will also go into the electronic lab book, which is of course now the newer step into how documenting your results. But if we keep it to the classical lab notebook, it should be bound, sturdy, high quality paper and numbered pages. So in principle, all labs have this type of books available for you. You have to make, or it is recommended to make an index table of content in the beginning of your book, so that for yourself, but also for people after you that are reading or have to find something in your books, it's easy to find. So index and table of content. Writing, as already Martina told you, must be easily legible to everyone. So English and full sentences. We had Spanish people in the lab before, I mean, years ago, that wrote their book in Spanish, and that was rather difficult and later to really find data back or to know what they actually have been doing. So English is of course a language that we all speak and understand, and that should be that is necessary. Very important, and also a lot of mistakes are made in that, is you need to write in a permanent ink, no pencils, and corrections should never be erased, right? It only should be crossed out, so that people, or if there is a discussion on who was first in a certain finding, or in a certain experiment that led to a certain finding, that they can be tracked back. And if they find some things that are erased or papers that are actually taken out of a book, then of course this will reduce your believability. And that could also lead to the conclusion that there are problems with the data or the time point where you actually found a certain result. So therefore, don't remove pages or leave open spaces in your book. All these open spaces should be filled up with a line through the blank space. For instance, in my lab, I check all the books regularly. And if spaces are open, I will myself personally actually put a line in this space, because it's of course very easy to put extra data or extra things in there, and that is of course not allowed. Right, so just very briefly, what is important when you write, or when you write down a specific experiment or the results of a specific experiment, write down something that you and others can understand, and actually see a kind of a logic path in a certain result that you finally have. So you start with a descriptive title, right? For instance, measurement, micro-CT measurement of this or that knockout mouse, right? Date of entry, very important. What is the purpose of your experiment? It's not trivial. For you, maybe yes, but for others, years later, not. Details on the methods, or a reference to what kind of methods you have used. Is that already in a database or as metadata somewhere? That is all fine. Include all the necessary details on the obtained results. Of course, now some of the results are huge files, fax data, RNA sequencing, or whatever. But then you have to make sure that people, even 10 years later, can find them with that's based on a certain name, as already Martina explained how to put that together in a server, in a folder, but of course not only on my personal computer, because if you leave with your personal computer, that's done. So indeed, try to put it on 3 different formats: personal computer, on a USB stick, and in a cloud or something like that. That's always the best practice that you can do. And of course, a conclusion. And it's not a publication. Really be frank. Say it didn't work because blah, blah, blah. Next time we should include this or that or that. Or it is as expected, and hallelujah, this is exactly what we needed for the paper. Right, it's all fine, but describe it so that people can also know that if something went wrong, that they can also learn from that. And very important is also sign relevant pages. I don't say every paper in that lab book, but putting in a signature and the date of a signature. And I also, for instance, write read and understood. I go through these books so that later, if there is a discussion, that there are at least 2 people, a PI or a group leader and yourself, that have verified, or that actually can people can go to ask for questions or anything that relates to this data. Right? Okay. So this is how a lab book could look like, very classical, but and maybe trivial for some, but I say that most of the people really have to, I mean, also when people come to our lab, they get an introduction in how to have to write or in that sense, data or their results, so that they can find them back and others can find them back later. So maybe a short note on the electronic lab book.
Okay, yeah, in our lab, we're making now an effort to change to an electronic lab book. Why did we choose for this? Mostly actually because it's not so easy if you've been in a lab for a long time, and that might not apply to if you just start out and are in your first or second year. But if you've been in a lab for 10, 15 years and you've had several students come in and out, it's not always easy to find a protocol, for example, that was done 8 years ago by some person. Then the person might have, I don't know, 5 lab books, and then you have to go to the lab book and really find the exact method. And that's, I think, very complicated. And for that reason, we have chosen now to go to the electronic lab book version. And I mean, this is really in its infancy, at least in our lab. So at the moment, we're still setting up databases and everything. And we will only probably start next year. But I think the electronic lab book is a good way, first of all, to organize your stuff, for others to read it easily and to find it. And one of the main benefits, I think, is by using these tags. So here I just copied like one very brief entry about a cell culture protocol. You can put in here how the, how the treatment layout was. So I think that's also quite handy, that you can just have these, you have these formats already for different well plates, and you just can put in your treatment and the N number and everything. And what I hope for the future, what will be very important, is that you can add these tags. So you can say, okay, this was iron treatment involved. You looked at BRDU proliferation at cell culture. This is the substances you use. It's mouse osteoblast. So of course, these tags also have to be defined in such a way that everyone uses the same words for, or tags for like mouse osteoblasts for a specific material, let's call it. But with these, later on, you can really filter all the entries of the lab book. And so I'm hoping that later with these tags and search results, that you can then more easily find a specific protocol that, or maybe even result that you're looking for. I also see that Andy is here. So Andy is also in one of our core groups who's setting up this electronic lab book. So maybe he also wants to make a comment on this, like later on in the questions and answers section. Because I think here these things are, like, you can put in your protocol, you could put in a Western blot result, the PCR result, so you can just drag everything in quite easily. It's not really meant for storing a lot of data. So again, for data storage, it's better to have your own platform, which for us is the T server, but I don't know, I guess the CRTD and all other institutes probably also have their common server where lots of data can be stored, but you can put the link in, data to be found, and then you just put the link from your file or folder structure in the end. Or what is also quite nice, I think, is that you can upload, let's say, your antibody list, have all the data sheets with the antibodies, and then you could just make a link, use this antibody in this concentration with this blocking agent or something, and then you can immediately go to the homepage of the provider of the antibody, for example. So yeah, I think Andy, maybe, as I said, maybe later you can comment a little bit on this, how you find it. So as I said, we're just establishing it. So maybe in a few years' time, we can tell more experience about this. Should I continue, or yeah? So another electronic database that we're very fond of is Pirate. Probably every one of you who works in Dresden knows this one. It's a database for managing your mice or animals, and it's, I think, also, yeah, very handy. You can put a lot of information in, and you can really track one mouse with great detail. So for example, here you have your mouse identifier number, then you see exactly who the parents were, cage number, where it is, the sex, the strain you're using, mutations, age, date of birth. You can put in comments. And again, now let's see, okay, you found lots of cages, you have many different strains in your lab. but then you can say, okay, I want to pick one specific mouse line that I want to look at, and then you just see these ones. And then you can also, by like clicking here on the sex, for example, sort that you have first all males and then females, or you could sort by age if you want to see the younger ones first and the older ones. So it's a very easy tool. You can put all kinds of treatments in, you can put the animal license numbers in. So theoretically, this is really a good way to document stuff. And similar to the electronic lab book, also in this database, you can't really undo entries. So you can only correct them, but you can't erase them or anything. So it's similar again to what you're supposed to do in your written lab book. You can't change anything that was already there. In the electronic lab book, actually, also there's a mode where the PI or some other person at some point says, okay, this is the final entry. And then the system also fixes that. And then you also can't add any information anymore or anything or change anything. Yes, okay, last part about scientific misconduct.
Yes. So, as you probably, I mean, as is of course also discussed in very extensively in the good scientific practice, is misconduct, and of course the integrity that scientists, or that we all need to have to actually bring those data as we analyze them, or as we in the experiments we obtain. And of course not manipulate them so that they sound really nice in a paper to get a high impact factor paper. You can find on the net whatever you like with regard to fraud, which is misconduct. I'll give you a few examples that might sound very obvious, and then later we have also an example that we actually use in our workshops that might induce a discussion. So there is at the EMBO Press, there is what I call an image detective. It's Jana Christopher, and she gave a very clear example of what fraud and misconduct might look like. And in one of the manuscripts that was sent to them a couple of years ago, this kind of Western was displayed, figure 1A, with these bands. And she already saw in the beginning that, or when she was looking at it, that there was something wrong with this Western. So she contacted the authors and she asked for the original file. And after a couple of days, the authors sent her the original file, and it looked like this. So this is actually an oversaturated blot, where they actually took the oversaturated band, or that what you see here, as the let's say to quantify or to show that these two first bands are the ones that are the most overexpressed, or are overexpressing the gene or the protein of their interest the most compared to everything else on that gel. She was still not satisfied, and what she actually found, or said, that it is a fabrication of an original Western that she finally didn't get from them, of course, because two of the, so here on the left hand side, the red part, and the other on the right hand side are exactly a copy of each other. So it was actually a Western, or like fragments of different Westerns that were put together just to fabricate a kind of result that looked like what these authors wanted to publish in this manuscript. Obviously, this manuscript was rejected. There is and I don't see it now here, but underneath you will probably see it. There is a website that I actually got from Martina a couple of years ago, where you actually drew my attention to, because there were some issues in the field that we are working in the hypoxia field, and I will come to that later. But this was also a very clear example, and it's pretty recent, like two years ago, in a Nature paper, where people, I mean, and the data, I just put them all. This is just a fraction of all the data that has been manipulated in that paper, starting already in figure 1I, namely that the ICAM and the VCAM is actually a mirrored copy of each other. right? And then going to all these different plots and mice, westerns, fax plots, you know, and these are different copies of each other. And I mean, it's obviously for everyone, it's obvious for everyone that this is of course not done. But you could also say, like, how stupid can you be, right? So of course this is always the tip of the iceberg, but shows that that people are really doing these type of things to get into a journal with high impact, with using this type of misconduct, right? This is then the person that I know personally from my field, Janine Erler. She's a Danish scientist, was a big or a hot shot, let's say, in science when she was in the States in the early 2000, 2005, 6. Then came back to Denmark and put up their company. She was working on the lysyl oxidase enzyme, and they made even inhibitors against that with the company. Finally, this company stopped using or developing this as a potential therapeutic drug because it really didn't work. And then just before that, Janine sold that company for big money, and then it was actually done with everything. And what actually was found out later is that, and this is just one example of one paper when she was in during her postdoc, is that she has fabricated systematically, let's say, right? So data, and this is just a simple example here of putting Western blots, cutting Western blots here and gluing them together, whereas the household protein here was on another blot. So you might think, oh, this is a small thing, but it is really misconduct. Just a small example, because she has done many more things wrong. Also, her husband, who she now is divorced from, had also a lot of his own papers and from his own group that that contained a lot of misconduct. Still, I'll go there. She got an ERC grant a couple of years ago, and still with the data or with the results and the accusations here and the proof actually of her misconduct has not yet led to really retraction of these papers. Now I think the 2004 paper is retracted, the one with Amato Jasha. But as far as I know, and I didn't check it actually lately, but she still has her ERC grant. And she's still in Denmark having her own group. And I saw her I still saw that she was presenting at meetings. So although of course, her integrity is completely lost for a lot of scientists, she's still in the business, which is of course not good. Then actually seeing these examples, of course, are very clear and very absolutely lack of integrity. We come to, or that is something that we also learned in the workshop, is when we have the following, let's say, case. And it's the case, and I will just briefly explain the case here. It's a PhD student called Alan Jagers, and he's, let's say, 1 year in the lab, and he's guided by a postdoc there. And he's doing some receptor binding assays and in vitro assays where he is using a certain cell type, and he looks at the cyclic AMP binding depending on incubation times. And as you can see here, he has done 6 different experiments, right? 6 independent ones. And what you can see here is overall in time, there's an increase of the binding. But as you can see, there is a lot of variation between the different samples. So it's next week, it's his, he had to, or a week after that, he had to present for the lab, and he shows the data to his postdoc who is guiding him, and he says like, okay, this is what I have. it's not significantly different. So I think we don't see that what we really wanted to, or what the hypothesis was. And the postdoc says like, yeah, you know, I mean, John, which is the PI, he doesn't really like this type of data where here in the lower part, this is probably cells that did not work or that died. So he asked Alan, so do you know whether they died here? Alan said like, no, I didn't have the impression. So the cells looked good, but they didn't give any result. And also the postdoc says like, yeah, this data, we normally take them out because that's probably an outlier. And so by cleaning up this data, he said, we probably can get to a significant difference in this process. So let's present it like this. And so Alan is just a one-year PhD student. He's confused and he says like, but this is, I'm going to manipulate the data here. Is that correct? So that's actually where the discussion stops and postdoc PhD student goes away. And so Alan is really confused in how he should now proceed. So the question that normally when we are all together in one, let's say, in one room would be like, how would you react to this opinions from the postdoc and the PhD student? So if any one of you has a good idea here, you can bring it up. Okay, so if not, then the point here is that you should never, especially in these settings where you as a postdoc, for instance, are teaching PhD students and how to present data to your PI or anything, actually force someone to let certain time or let certain measuring points or certain experiments being taken out of a complete set of data without any specific calculation of one or other data point being an outlier. You cannot just say because it lies out of everything that is shown here in this graph, unless you really have statistical data on that. And if these confirmed by the PhD student are not a bunch of dead cells starting at the experiment, you cannot just erase or let these results just not be part of this entire set. So in principle, the point of bringing this thing is that you should not change data by making them just nice, right? So handle all adjustments with care and keep it safe and communicate with everyone who are actually part of the experiments and part of the whole project to decide on how to proceed, right? And the best proposal or the best suggestion would be here is to at least repeat it once, twice, or 3 times more to make sure that you go in a certain direction, or that how the experiment has been shown by 3-4 of the experiments are then can be confirmed in that way. So don't erase data that statistically should not be erased at all. So the take-home message for this is that you always have to think twice when working on your data, right? And this is just a simple example of a binding assay, but it is also clear for immunohistochemistry, for instance, or anything else where certain manipulations or certain adjustments can already be easily interpreted as a manipulation. So there is also always a limit to beautifying your data and to changing stuff. And that is what already Martina said, it all also depends on the rules of the journals. What can you do? What are you allowed to change or to adjust? Also, depending on your research niche, of course, on the guidelines that belong to your scientific discipline, right? We have other scientific guidelines or other guidelines than other disciplines, for instance. But that is always important. And most importantly is communicate with a lot of people inside your group. So, and in case of suspected misconduct, and this happens more than you think, or misconduct by others, you can always contact, and that is also something that Martina already brought up, person of trust or ombudsman, local ombuds people, local persons of trust in CRTD, MPI, or in the MTZ, or in the Uniclinicum. And of course also university or even the DFG. You can always contact them, and they will always help you out with your concerns. So publication misconduct, something that always is an item and is also a part of good scientific practice. Fraud, obviously, as we said, duplication of publication, plagiarism seem very trivial, but they always come up. Omitting sources is, of course, one, omitting references, right? Gift authorships, which is also part of good scientific practice. Denying authorships, we all know about that. Acknowledgements, do you acknowledge certain people that have done something but are not necessarily part of your authors? And of course, review, sabotage, and this is, I think we have all massive examples of these things. This is, of course, one of these important pillars of good scientific practice. How do you treat your other the other scientists that you are reviewing? Right? Very important. So co-authorships, as according to the TU Dresden, and you can find these recommendations in that pamphlet. So co-authorships are not established by just acquiring funding, and just is, of course, I don't mean it in that way, but acquiring funding for a project that leads to a publication is not or should not be enough to be a co-author. That can be, of course, discussed, and you can, this can be openly discussed here. Providing standard investigating materials should also not be a reason why you should be a co-author. And with standard methods, we mean give you an advice on the melting curve or in a certain PCR or so. But of course, giving a certain cell line that is not available, or a knockout mouse, that is of course a different issue. Also, only technical participation in data collection is or does not have to require or lead to a co-authorship. And that is, of course, all definable by people that are doing these experiments or that are doing making the publication. Technical support only, then it means merely providing equipment or laboratory animals that you can get from everywhere. That should not be enough. Allowing the use of data, but small amount of data, of course, reading the manuscript without making substantial contributions, or leading the department or a working group in which the publication has originated is not necessarily enough to actually lead to a co-authorship. Of course, again, and this is very important, these are not like rules or laws, these are recommendations, and they, of course, should be discussed with among each other with people that are part of the publication that will be, of course, published. So to come to the end, let's say, I think the take-home messages from us is try to develop your own scientific integrity, right? That is not always that common, and you just have to analyze it. You have to think about it. How do you deal with results? How do you deal with other people? How do you deal with data from others? How do you react to critique? These type of things. Think about it and build up your own scientific integrity in that. That will be helpful for yourself, but also towards others, be very helpful in your career. In case of a conflict, get professional help, talk to people that you trust, go to your local counselors. That is an advice that a lot of people take. We see a lot of people, and that is fine, that is good, because before or 10 years ago, that was this bridge between people that have questions, problems, and people that could be helpful, or PIs or other PIs there was no bridge, and this is made in the last couple of years, and use that. If you think like I have an issue, I have a problem, then try to find people that can help. Right? Know your rights, very important. You have rights for using data, obtaining results, but you of course also have obligations as a researcher towards other people, towards people that you guide, and that's also something that you have to realize. And preserve your data in a secure and correct way. I think we have given you enough examples here. And communicate with your guides, your mentors, and your PIs. Right, that is very important, so that you learn and you also actually make or develop your own scientific integrity there. So thank you very much for listening. One small note here is try to do always the right thing, even when no one is looking, and that is absolutely important in integrity. Thank you.
Okay. With this, I think we will stop the presentation, and I'm sure there are lots of questions on this very important topic. I assume everyone has already made some experiences, or yeah, so any questions? Don't be shy. Andy, do you want to share your experience with the electronic lab books so far?
Hello, everyone. Can you hear me? Can you hear me?
Yeah.
Yeah, okay. Yes, I can give maybe my personal experiences with the lab folder software that I'm just currently testing for the moment. I find it very useful because you can really, as Martina has already presented, you can really put in everything. You can cross-link. So for example, I'm writing now, okay, especially maybe as a suggestion also for the PhD student, you would start with one project. You come to a new lab, you have one project, and you might think you have really the overview, but the more things you will do in the lab, the more things will pop up. You will have new ideas for new projects. Then you will maybe have also a plan B project if plan A is not working. And to really structure these different projects, it's really that important to really save the data, to document everything. And for my experiences, comparing the normal lab notebook versus the lab folder, I really find the lab folder really more useful, because you can really search for different key tags, for example. Also from my experience, when you work with a technician very closely, so I have one technician who is doing a lot of cell culture, the documentation is quite easier, because then I do not need to give her or my students the written lab notebooks, and then they have to search for something, because even I would forget everything that I did in 2013, for example. And if you would just search for text, then it's really easy. And also what is helpful, that you can cross-link different links. For example, you would work with a specific kit in your lab, then you can cross-link the homepage maybe from the company where you find the protocol. You have a direct access. Or for those people who work with cloning in the lab, you can directly put in the picture of maybe the vector map. That is really helpful. And yeah, so from my side, I'm very satisfied with lab folder.
Thank you.
Are there other people that have another experience with regard to lab book or electronic lab book, or a version that you like?
I just have a question with the electronic lab notebook. In the case that you want to, I don't know, modify something from the past that, for whatever reason, not to change that or something, there is a way that everything gets documented. Like I did this on, I don't know, 2019 and 2020, I modified this kind of data protocol or something. Is that secure, or you can erase past data and forever or something like that? Do you understand what I'm trying to say?
So if you can fix, so in general, if you make an entry, you can still add or change things in a way that it will be documented. So I mean, you will still see your original entry, but you can still change it. But once the PI signs it, so to say, in the electronic version, then it's fixed, and then you also can't change anything anymore. I guess then you would have to do maybe like an additional tag on if you really needed to add something. But I guess then the question is, yeah, at some point, you like have to fix the written documentation already at some point and say, okay, this is how it was. So I mean, in 1 year time, you won't know what you did back then in that much detail anymore. So
and with this tag that you have, probably you can link that to newer forms of that protocol.
exactly,
yeah.
So yeah.
Okay, thank you.
Okay.
Anyone else with other questions or remarks or things that they want to discuss?
Yeah, maybe I can add something as well, because you also said, Ben, that you, whenever someone new is coming to the lab, you always make this introduction how to document the things, right?
Yeah.
I think that is very, very important for all those, even if you are a PhD student and there is like a lab rotation that you get, and this is your first supervision, always at the beginning really have to communicate with your student, show him or her really how to document what is important, that they really get like an idea what is important, you know, because sometimes when I remember also my time as a PhD student, you start very naive with some things, and then you get a feeling what is really important, right? And that it's not really that trivial. So always communicate, or even with a technician, because very often I had also the experience that the technicians are also very interested in the projects, and sometimes, so I always, when I speak with my technician and I explain what she can do in the cell culture, I always explain why she is doing this. So what is the purpose of the study? Why is it important to treat the cells with this over that? So that they also get a feeling when they document the things for themselves, okay, maybe that is important to document this aspect, even if they observe something that might be not important, but when they know, it could be important because the purpose of the study is this and that they write it down or that they communicate it with you. I think that's really important to communicate always.
Yeah, absolutely. Yeah.
Nothing to add. Communication is definitely key. Yeah. Okay, I think if there are no more questions, we'll just end this here. So I think Ben and I think you can find his email or mine as well. If there are some more questions, if there's something coming up afterwards that you want to ask, so that's no problem. And otherwise, I think if there are no more questions, we'll just close this here. Say thank you for listening, and happy documentation. Okay.
Okay. Bye-bye.
Bye. Also wait to the camera. Bye.
Bye. Thank you.